Buenas tardes. Se abre la sesión de la Real Academia de Ingeniería que tiene por objeto proceder a la imposición de medalla al académico correspondiente don Samuel Stupp. En los estatutos y en el reglamento de régimen interior de la Real Academia de Ingeniería se contempla la figura de académicos y académicas correspondientes que serán elegidos y nombrados entre personalidades no residentes en España que se hayan hecho acreedoras a tal distinción por méritos análogos a los requeridos para ocupar plaza de académico de número. Estos requisitos son: se requiere una cualificación relevante, además de una reconocida integridad profesional, así como reunir alguna de las condiciones siguientes. Haber realizado aportaciones de destacada importancia en materias teóricas o aplicadas vinculadas a la ingeniería que estén reflejadas en patentes o publicaciones de pertinente nivel. Haber demostrado excepcional competencia para lograr avances significativos en nuevos campos tecnológicos o en su desarrollo empresarial. También constituirán méritos adicionales para la incorporación a la academia el haberse mostrado eficaz en la dirección de empresas e instituciones, poniendo de manifiesto capacidad para afrontar y resolver problemas nuevos o complejos y contribuir a la innovación, así como haber prestado servicios relevantes para la docencia y formación de ingenieros. Tiene la palabra el secretario general, el excelentísimo señor don Eloy Álvarez Peregrí.
En sesión plenaria de la Real Academia de Ingeniería, celebrada el 28 de abril de 2015, fue elegido académico correspondiente el ingeniero don Samuel Stupp. El profesor Samuel Stupp obtuvo su Bachelor in Science en Química en la Universidad de California en Los Ángeles y su PhD en Ciencia e Ingeniería de Materiales en la Universidad de Northwestern. Su investigación se centra en materiales para la energía y la medicina, cubriendo los campos de autoensamblaje, materiales supramoleculares, electrónica orgánica y materiales biomiméticos.
Tiene la palabra la académica excelentísima señora doña María Vallet Rollí.
Excelentísimo señor presidente, excelentísimas señoras y señores, querido Sam, queridos todos. El profesor Samuel Stupp nació y creció en Costa Rica y llegó a Estados Unidos en 1968 para iniciar su formación universitaria. Obtuvo su licenciatura en Química en 1972 en la Universidad de California en Los Ángeles y su doctorado en 1977 en Ciencia e Ingeniería de Materiales en la Universidad de Northwestern. Se incorporó al cuerpo docente de Northwestern tras su graduación en 1977 y luego se trasladó a la Universidad de Illinois en 1980 y regresó a Northwestern en 1999. Actualmente es catedrático del Consejo de Administración de Ciencia e Ingeniería de Materiales, Química, Medicina e Ingeniería Biomédica de la Universidad de Northwestern. Dirige el Instituto Simpson Quarry de la Northwestern y el Centro para la Ciencia y la Energía Bioinspirada, un centro de investigación de las fronteras de la energía financiado por el Departamento de Energía de los Estados Unidos. El profesor Stupp es miembro de la Academia Nacional de Ciencias de Estados Unidos, la Academia Nacional de Ingeniería de Estados Unidos, la Academia Nacional de Inventores, la Academia Americana de las Artes y las Ciencias y es correspondiente de la Real Academia Española de Farmacia y ahora se incorpora a la Real Academia de Ingeniería. Es miembro de la American Physical Society, la Material Research Society y la Royal Society of Chemistry. Entre los premios que ha recibido se encuentra el premio del Departamento de Energía por sus destacados logros en química de materiales, el premio de la Sociedad de Investigación de Materiales, el premio de la Sociedad Química Americana en química de polímeros, el premio Ronald Breslev de la Sociedad Química Americana por sus logros en química biomimética, el premio internacional de la Sociedad de Ciencia de Polímeros de Japón, el premio de la Real Sociedad en Soft Matter and Biophysical Chemistry y el premio de nanociencia 2020 de la Sociedad Internacional de Ciencia, Computación e Ingeniería a Nanoescala. Ha recibido doctorados honoris causa de la Universidad Técnica de Eindhoven, los Países Bajos, la Universidad de Gotemburgo, de Suecia, y la Universidad Nacional de Costa Rica. Samuel Stupp es un químico y científico de materiales reconocido por su trabajo sobre materiales supramoleculares, autoensamblados y biomateriales bioactivos. Ha desarrollado estrategias para crear polímeros supramoleculares y materiales híbridos orgánico-inorgánico, con funciones relevantes para las energías renovables, la electrónica orgánica y la medicina regenerativa. Un trabajo muy reciente se centra en la dinámica supramolecular para crear materia blanda robótica y optimizar la bioactividad, así como el desarrollo de materiales fotosintéticos. Stupp tiene conexiones profesionales con España desde hace muchos años y actualmente es el presidente del Comité Científico Internacional del Instituto de Bioingeniería de Cataluña, IBEC, y forma parte del Consejo Científico Internacional del Biomagune de San Sebastián. Samuel Stupp está considerado como un líder a nivel mundial en el campo de autoensamblaje de materiales. Se le considera el primer científico de materiales que introdujo estrategias de autoensamblaje de materiales orgánicos en 3 dimensiones y no únicamente en superficies. Y para mí es un privilegio introducirle como académico correspondiente de nuestra Real Academia de Ingeniería. He dicho.
Tiene la palabra don Samuel Stupp, quien pronunciará una conferencia con el título Self-Assembly of Soft Materials for Advanced Technologies.
Muchísimas gracias, María, por tus palabras y también al doctor Colino y al doctor Álvarez. Me siento sumamente honrado de tener este privilegio. Es un gran honor para mí y quisiera agradecerles por la oportunidad que me han dado, el reconocimiento y mis mejores deseos para la Academia Real de Ingeniería en el futuro, y espero tener nuevos contactos con ustedes en los años que siguen. Dentro de unos minutos vamos a cambiar de lenguaje, de lengua. Vamos a cambiar al canal de inglés, pero también quería decir antes de comenzar la conferencia que para mí esto también es especial, obviamente por mis raíces iberoamericanas, habiendo nacido me crié en Costa Rica, viví allá 17 años y por lo tanto mis conexiones con España tienen mucho significado personal. Esta es otra dimensión de este nuevo contacto. De nuevo, muchísimas gracias por el reconocimiento y el honor. Okay, so now we turn to English, and I'm going to talk about self-assembly of soft materials for advanced technologies. Okay, so vamos a ver si tengo sí, aquí, perfecto. Okay, so the first thing I'm going to mention is just provide a pictorial view of what we mean by self-assembly and by supramolecular materials. And so as the slide depicts, in self-assembly, what we look at is the organization of molecules occurring spontaneously as a result of very specific interaction into a structure. So when materials are designed for a specific function this way, we refer to them as supramolecular materials. The slide shows at the left-hand side 4 publications from our laboratory in this field as we started that journey back in 1993. So, a few comments about soft materials through molecular self-assembly. So this is basically a field that is giving rise to an emerging form of soft matter, which is not necessarily the traditional polymers that we know, the covalent polymers that are so important in technology, but it is a completely different approach to creating organic soft materials. And just a bit of background, I mean, this area emerged as a field in science and in technology as a result of the great interest that there was in biological structures. So, for example, in the 1930s, people thinking about beta sheets in peptides, in proteins, alpha helices, DNA double helices in the 1950s. This is the backdrop to the creation of this field, because people were, in those decades, interested in understanding biological structures that were judged to be critical to living processes. And all of those are indeed self-assembling structures. All of those are indeed supramolecular materials. And so this is then the way that this field began. So it begins to blossom in the early 1990s. And so, as I mentioned earlier, in 1993 was when we published the first paper on the subject. And at the very late times in the 1980s, 1987, there was a Nobel Prize in chemistry given to Jean-Marie Lehn and two other chemists that recognized the new wave or the new interest in science in understanding very specific interactions among molecules. And so that also was in the backdrop, in the historical backdrop of how this field developed. And now supramolecular materials then are materials that in which you have assemblies which may be finite, but they are formed by thousands, millions, and billions of molecules connected into a well-defined structure. And this type of assembly occurs across scales by many mechanisms, which in chemistry are recognized as, for example, hydrogen bonds, the so-called pi-pi overlaps, which have to do with the overlap of wave functions of conjugated molecules, and steric forces, hydrophobic collapse, when molecules find themselves in water but are actually hydrophobic in their chemical structure. There are processes of collapse which lead to organization of molecules. And in all of those, I would say, both enthalpy and entropy play a very important role. So one of the interesting things about, or one of the unique things about supramolecular soft matter is their dynamics. Because the molecules, in contrast to normal polymers where you have monomers that become connected by covalent bonds, which are very difficult to break, very strong bonds, in supramolecular materials, the monomers, and biology does this all the time, and sometimes the monomers are actually proteins, large proteins. So the monomers connect to each other by weaker forces, secondary bonds, that are possibly 10 times or more weaker than the covalent bond. So what that affords to the material is a special type of dynamics that are not observed in other soft materials. So this is a very exciting way to think about new forms of soft materials. And one way to think about it is that the bond lifetimes are finite. For example, hopefully they're less than 60 seconds, the lifetime of a bond between monomers. But it should be at least, it should be greater than a microsecond. Otherwise, you don't have a material. You don't have a solid substance that you can take advantage of in a mechanical sense or in a thermal sense. So the bond lifetimes are finite, and they are possibly in a range larger than a microsecond but less than a minute. And so the bonds are not permanent. Okay, so beyond just the structural programming, which is one aspect of the field, meaning that you can design molecules and create a structure that has a function. Understanding, so we are learning a lot about that, but we are just beginning to learn now about supramolecular motion. So these materials basically internally have a life of their own. You can imagine molecules jump around a lot, move from one place to another in ways that you don't expect other materials to do, because they might be a static crystal, for example. So molecules have a lot of motion, and there is a myriad of motions that we still don't understand exactly or are to be discovered, types of motion. So this is very, very exciting. And that's one of the areas in which my lab concentrates these days. And in general, our targets in terms of function in the materials that we design are advanced medicine. We want to use them for advanced medicine specifically, we are interested in regenerative medicine, but we're also interested in other therapies. We want to use them in the problems of renewable energy, which are so critical to the planet. And we want to make a contribution with supramolecular materials for renewable energy, and also for problems of sustainability and environmental problems as well. So those are the areas in which we concentrate. And so now you're going to hear, some stories, and I'm going to not describe all the details on the slides. There's a lot of detail there, but just so that you get a feel for what these materials are like. This, I can point there, or would that work? No, I guess it doesn't work. I'll use my pointer here. So this is one of the most interesting structures that I consider a very important discovery, for in our work. And this is a paper in Science in 1997, where we designed this molecule, which looks, that you see on the slide on the left side. It's a molecule that is kind of large as organic molecules go, and it was pretty challenging to create it. But the interesting thing is that molecule, which is ordinarily you wouldn't think it's an important molecule, self-assembled spontaneously into this mushroom-shaped nanostructure, which measured, let's say, nanometers in its dimensions. And then the mushrooms stacked in a way that a mushroom farmer would have never picked when packing mushrooms in a box, right? Because they packed from stem to cap, stem to cap, stem to cap, where the most logical thing would have been to basically position them in anti-parallel orientation. Well, for enthalpic and entropic reasons, and we have published a lot about this, they chose to be parallel. And so what this created, because the mushroom doesn't have inversion symmetry, and as they stacked over many, many layers to produce a macroscopic film, a transparent macroscopic film that to your eyes, if you would have seen it on a table, it would have looked like a coating of polystyrene, a transparent plastic, and there wasn't a single polymer molecule in that film, but it looked like a plastic. So that was pretty exciting. The orientation of the mushrooms, then what it did is it spontaneously created a piezoelectric film, which meant that when you press on it, it produced an electric current. So that was, super exciting. And it also created a film that would always have one side hydrophobic and one side hydrophilic, no matter how thick it would be, because of this polarity. So this was a very exciting discovery. In a more recent example, which I will show you here, I worked with my colleague Fraser Stoddart, who won the Nobel Prize in Chemistry just a few years ago. Our two labs worked together on this project, and we designed, because we brought different types of expertise into the problem. Very short story here is that what we created is these crystals made from these very small molecules, made of electron donors and acceptors. And when they stacked, basically blue and red, they have charge transfer, and then when an electric field is applied externally, the direction of polarization can be reversed. And so, in fact, this is what is called in science ferroelectricity, which is an extremely important phenomenon for memory. And people are hoping that this kind of phenomenon from organic materials will be someday useful to store information. And there's some aspects of it that are important in quantum computing as well. So this was exciting. Now, what is the fascinating part about this? If you look at the drawing on the left, you see the red and blue molecules are connected by these dashed lines, which is a very specific hydrogen bond. And it is that bond, precisely, that created a ferroelectric material at room temperature. So in Japan, a group at the University of Tokyo had discovered that they could mix molecules like this and create ferroelectrics, but the ferroelectricity was only possible at 85 degrees Kelvin, which of course would make it meaningless in a technological context. And that simple hydrogen bond stabilized the ferroelectricity all the way up to room temperature. So this was an important thing. Another example are these systems which I have spent now 20 years studying, and these are the materials that we called peptide Amphiphile supramolecular materials. They're made out of, they are fibers that are formed by specific molecules that, as you see on the slide, self-assemble in water to create a fiber. And that fiber sometimes is cylindrical, or as you see down here at the bottom, has the cross-section of a football stadium, okay? Or sometimes it twists, as you see in the micrograph that's in the middle of the slide. These filaments create materials that take the form of hydrogels, or create, or also just soft materials, which are not necessarily hydrogels. And my lab has probably produced upwards of 500 or 600 different forms of these materials for many different functions. And this is going to help me explain with these materials this issue about the dynamics, which is so interesting, and also how we use them in medicine. So the molecule, you see it at the top, this is the monomer, and the monomer then has a peptide sequence, and this peptide sequence when the self-assembly occurs, if you put a signal here, any kind of signal at the very end of the molecule, then you can display that signal in an incredible density, upwards of 10 to the 15th power per unit per square centimeter. So it's very interesting strategy that can create highly bioactive, if you put a biological signal, but you could put a chemical signal or anything else that you want. You see them here by cryo-electron microscopy. They are extremely long filaments, microns in length, sometimes macroscopic in length, but the diameters are only about 10 nanometers. So the aspect ratio is enormous. It's comparable to carbon nanotubes. Okay, so they're really interesting materials. They present signals, and they, if they are made from these monomers, they're completely biodegradable. They would biodegrade very fast, either in vivo or even ex vivo. They would degrade fast because they are made of natural structural units, amino acids, lipids. We use sugar moieties to construct them. We use nucleic acids and various things of that sort. From a dynamic point of view, this is an interesting slide. And so you are looking now sort of at a dynamic map. So you see red color and blue color. And over on the left-hand side, you see that it is red on the outside where those signals are. And that means that on the surface, these structures behave like a liquid. And we actually measured this by a technique called electron paramagnetic resonance. And we found that the motion of the red part is comparable to a liquid in the nature of the motion. As you go down 1 nanometer or 2 nanometers into the core of the filament, it turns blue because there it is a solid. And if you keep going another few nanometers to the very center, it's kind of a pinkish color, not quite as red as the surface, because in the middle is more like a liquid crystal. So you have 3 forms of condensed matter in the space of about 7-8 nanometers. It's really fascinating, to see those changes that way. And so this is one of the first things we saw in the dynamic department in 2014. In this paper here in Science in 2018, I'm going to pick up a little bit of speed because Maria warned me I didn't have hours, but I will do my best here. This work in 2018 really proved to us that they can have extreme motion, the molecules inside these structures, but at the same time, it taught us that we can tune this motion so that we can design specific properties. And that's a very exciting direction. How do we discover that? Well, you look at 2 filaments here, blue and red, and on the surface, we put DNA. We put a single-stranded DNA sequence, and the red one and the blue one are complementary, which means they do the Watson-Crick pairing that happens in our genes, in the nuclei of our cells, where the nucleotides recognize each other. Well, this is the same phenomenon. They're complementary. And the reason we did it is because we wanted to find supramolecular ways of joining the filaments to create perhaps more complex structures. And also to make them reversible, because you can disassemble the Watson-Crick pairing. This is how genes are expressed, actually. When a gene is going to be expressed by a cell, you have to sort of unwrap the double helix so you can read the genetic information. Well, anyway, so we did this, and we made a discovery which is quite fascinating, and it is summarized in that slide. So we had only 1% of the molecules with DNA, and then the rest of the molecules didn't have DNA. They were just normal peptide amphiphile molecules. We found that within minutes, as you see at the top, in about 9 minutes, this very strong fluorescence appears in front of your eyes because we put a fluorophore in the DNA. So the fluorescence concentrates. Suddenly, initially you don't see it because there are very few molecules with DNA, therefore the fluorescence is very weak. But suddenly, in certain spots, the fluorescence is super strong, and you see it happen over minutes. And so what is happening is that the DNA-containing molecules walk. They escape from the structure where they used to be, and they concentrate in other structures. And so then certain parts of the structure are strongly fluorescent because they contain all of the DNA, and then in the rest of the sample there's no DNA. You see that very clearly here. You see that's a scanning electron micrograph at the top, and the purple, which is false color, those superstructures, which are bundles of hundreds to thousands of filaments, is where all of the DNA has concentrated, and then around it there's no DNA. And that means that molecules had enough dynamics to escape where they used to be, and then, I guess you could use the word, they colonized another assembly that was maybe tens or hundreds of microns away. That was very exciting, and in fact, you can see at the bottom that when we disassemble the DNA, which is what drives the formation of these bundles and this, what causes the molecules to walk, is their driving force to create this pairing among the bases. When you disassemble, as you see at the bottom, the purple regions are completely gone. By just putting a drop of a molecule in water on top of it, so that it will disassemble, cause the disassembly of the Watson-Crick pairing. And that has a mechanical consequence. So it's very exciting because you are looking at a soft material and you put a drop of water in what's originally a very soft material. After you put the drop of water, it becomes a stiffer material, or vice versa. Because you can use both types of you can go in both directions. So when you disassemble, the material becomes softer. When you reassemble the purple regions, the material becomes stiff. And so it's just an amazing thing, right? That you put a drop of water and then you reverse it, and then the material goes from soft to stiff, and then you put a different type of drop and goes from stiff to soft. I have no doubt that someone will figure out how to use this in technology. I mean, and it's a very exciting observation. This is what we did a computer simulation of this, and so you see it here. The computer simulation predicts the formation of the clusters. So you see there, the clusters form. This is what the simulation says. This simulation takes about 3 to 4 weeks, and we use 8 processors to do it. I mean, it's a big simulation. It takes a lot of computer time. And if you don't allow the molecules to escape, right, you would not see these bundles. But if you allow them to escape, then this is just a larger view of the assemblies. So if you allow them to escape, which you can do in the simulation just by putting in your force field, you put a strong cohesive force, and so they cannot walk away. And so if they walk away, they make bundles. If they don't walk away, they don't. So then that led us to create, and if anybody listening to me here or elsewhere on YouTube is interested, you can read this paper, and this map, this three-dimensional map basically is what we came up with, where we were basically able to say, if you have cohesion among the molecules in this specific range, molecules will escape and they will build superstructures when they are driven to interact with each other. If you are in a different energy regime, which you design through chemical structure, those superstructures will not form. So it's like a map. It's like basically it is a recipe to design molecules when you want them to escape and create superstructures, and when you want them to be static and not escape. So this was an extremely valuable thing, and so this actually gave us a very good idea, which I think you'll be excited to see it, because I haven't talked about this very much, and I will tell you in a minute why I'm talking about it today. So, hold on. Exciting. Okay, so I told you that we're interested in advanced medicine, and so because we want to use these filaments as basically an artificial extracellular matrix that signals cells for regeneration. I mean, so that's how we're using them. And look at this slide right here. This is scanning electron microscope images. On the left, you see at the time, my postdoc, Zaida Alvarez from Spain. Everybody calls her in my lab Zaida, which is proper, right? Except me, because being from Costa Rica, she's Zaida. Right. But my students think it's great because they think is a pronunciation of the TH, you know. Anyway, so Zaida removed the cells from the spinal cord and saw what you're looking at, this mesh of fibers. And then on the right, you see our material that we use, the type of material we use for regeneration. They look architecturally very similar. In other words, we are able to mimic very much the architecture of extracellular matrix. And we think that's why our cells really like our materials. In this scheme here, you see a fiber coming down. It's red, and it's got these signals in yellow, like little dots along the way. And those are the signals we put in our materials so that they can talk to receptors and cause something biological to happen. And we had a really great discovery in 2004 where with these signals, we were able to convert stem cells into neurons, neural stem cells, which can differentiate into different types of cells like oligodendrocytes or astrocytes, which are cells you find in the central nervous system. But here, our material and its signals was able to promote the formation of only neurons. So to this day, since 2004, this is, in my opinion, one of the best ways to make neurons. And so a lot of people are excited about that because people are looking to transplantation of neurons to address Parkinson's and Alzheimer's disease and other neurodegenerative diseases. So we have made a big contribution there, and we send our materials to many places for that purpose. This is a little bit of a parenthesis to the punchline, which I promised. So here is just another one, and this one is a material that's now formulated to have a slightly different, let's see if this froze here, but I will just explain, what this is. The previous one was just another material that makes bone grow. And it makes bone grow in a highly efficient manner. And let's see if maybe yes, I can do it now. You see it at the bottom. We're spreading, this is the spine, a model of a human spine, and this toothpaste-like material is coming out. This toothpaste-like material is now a hybrid that has a lot of supramolecular content, And it is a fantastic material to grow bone with very minimal use of growth factor. And so right now we are in a journey with the FDA in the US to try to get this approved for clinical trials, because it's a very good way to grow bone, and particularly is useful to do spinal fusion, which is a surgery that has become more and more common as we are approaching, this critical fact, right, that in about 10, 20 years, the population over 65, will be enormous compared to what it has been or what it is now. So this is a very important target. And you see here, we made it work in a pig, not just in a rat, and also in a rabbit. And so the spine of the pig is very similar to the human one in terms of size, and so we're very excited about that. Okay. So supramolecular dynamics. So you know now that we have these materials that can have great possibilities for regeneration. That's great. You also heard that we became aware that there is this fascinating dynamics that occurs with molecules escaping and blah, blah, blah. Well, so then we reason that between escape and moving, in highly localized way, the way you saw it in that map of dynamics, the red and blue map, there is a big gap. There's a big spectrum of motions. So, for example, what if molecules can walk within the fiber, without leaving the fiber, but just within the fiber? What if they can jump out, but then be able to change their minds and return back? It's a whole spectrum of motion. If they can jump out, right, then they can, it's almost like it's a material that is smart enough to stick its head out and stick, its business end out of an environment to signal something and then return. So in that dynamic event, there is intelligence in the material. And so why is it important to do it that way? Well, because of this movie. So look at this movie carefully. This represents a cell membrane, and those objects in green and blue are the receptors that we have on our cells, and those are the recipients of the signals that our supramolecular materials provide. So look how now the cells move. Cells move a lot. And so it's almost like biology understood that things need to be supramolecular and they need to move in order to enable life processes. And so the cell, you can imagine, is moving like this, right? The membrane is undulating and the receptors are moving. They are actually moving inside, anchored to the so-called lipid bilayer, but they walk. They walk and they go places. They don't stay static in one spot. So if you are a material trying to signal these dancing proteins and undulating lipid bilayers, you want a material that moves also, just like the cell does, so that it can fight to get more successful encounters for signaling. That was our idea. And so here is the exciting part. We made an amazing discovery that proved that this motion, that tuning this motion changes the way our materials signal cells. And as I was telling my colleagues here, my distinguished colleagues, were listening to the story about how I they canceled my flight today, and so I had to take another flight later. And so that was good though, because so I was at the airport, then boarded my flight, and just as my my airplane was about to take off in Chicago to Frankfurt, Germany. We could still have our iPhones live, and I downloaded my email. And our paper describing this was accepted in Science less than 24 hours ago. And I'm very proud that the primary author of that paper is a Spanish citizen. And that's the famous Zaida that we talked about. Zaida is the first author of that paper. And she was just an amazing partner with me for 5 years. And we did this over 5 years. So what is it that we did? Okay, so look here. It's pretty interesting. So this is now the motion thing as applied for, so hopefully this paper, you'll see it in about 6 weeks, I assume, but it's accepted just a day ago, less than a day ago. The material, the fibers, so we depict them here as moving. You see? They're moving. They're moving like crazy, right? And look, they can jump out and touch the receptors, right? They can, because the receptors are moving, they can jump out. They can get at the receptor from different angles, right? Because they can move. And our hypothesis was that this would improve signaling if we could tune just the right dynamics or the right motion. Well, the paper that's going to be published in Science will demonstrate that this works in vitro biology. So we actually did biological experiments to prove that it works. But the most exciting thing is that it works in vivo. And it works in vivo in one of the problems in regenerative medicine that I care most about, which is spinal cord injury, so that we can avoid paralysis on the planet, right? So that people that have serious accidents in explosions or gunshot wounds or skiing accidents, you know, take your pick, can be prevented from becoming paralyzed for the rest of their lives and be confined to a wheelchair. And this is, of course, a huge problem because it is also related to regenerative processes in the brain as well, because it's all the central nervous system where it is extremely difficult to do regeneration. I mean, it's the most challenging regeneration is in the central nervous system, the brain, which is what is going to preoccupy us for the rest of the century and beyond. This is like the century of the brain. And so it's a good thing that it's there. Now, I want you to see the in vivo biology. Okay, so I'm going to walk you through this because, you know, it's really interesting and I want you to appreciate it. So you're looking at 3 curves, and of course, you are all engineers, and so you appreciate curves and quantification, right? Okay. So you're looking at 3 curves, the purple, the red, and the black. And those curves just measure over a period of 12 weeks how the animal is recovering from spinal cord injury. Okay? So first, the black one, I'm going to show you the movie of the black one, which is the control. We just inject some saline after the animal has been paralyzed by a very severe injury. And we worked on spinal cord many years ago, on spinal cord injury, but at that time, it was a mild injury. This is a severe injury. This is a much more serious thing. Anyway, and so please watch the movie of this mouse on the right-hand side at the bottom, and that's the paralyzed animal. See? It just drags the hind legs. It's completely paralyzed. And so this is 12 weeks after we injured the animal, and we just injected as therapy a little bit of salty water. So the animal is paralyzed. Then in the red curve, we injected our supramolecular filaments, but we designed the molecules in such a way that the motion would be limited. So we call that slow supramolecular dynamics. So they don't jump around as much, or they don't walk as much. There's still lots that we, of course, won't know until years from now when we need new techniques. But now look at the animal. The animal, we're going to see the one that is treated with the slow moving. Oops, if the computer will let me. And so I think it'll work in a minute. There. See, it kind of walks a little bit, but not very well, right? See, it is able to put its feet down, but not very well. It is in a very awkward walking. And then the blue one is the supramolecular filaments where the molecules move a lot. They jump around and like there's no tomorrow, right? They move, and we actually, in this paper, we measure those motions with pretty challenging techniques. We measure them, and so we know that the blue one doesn't move, the ones in the red don't move as much, the ones in the blue do. Now look at the movie of the blue one, and just look at the animal walking. The animal walks pretty normally. The systems are identical in every respect. I mean, it took us 5 years to be able to produce materials that would be identical, except that one moves and the other one does not. Well, or one moves more than the other one. And then we created a material in between. The blue is not in this curve, but there is in the paper one in between, in motion, and it's in between the purple and the red curve. So we dialed the motions, and we see biology changing. And the paper proves it also at the level of histology, at the level, we actually observe that, for example, blood vessels grow when the molecules move, and when the molecules don't move, the blood vessels don't grow as much. The neurons myelinate when they move, when the molecules move, and they don't when there's less motion. The axons regenerate more after the injury when the molecules move. This is a piece of work that has consumed us, intellectually for the last 5 years because it's very exciting. I'm hoping that one day it will help humans that are paralyzed. So now you know what this is and how we use it. I'm going to mention just a few other examples of other things, and I'll finish up, so that not to extend or abuse my time here at the podium. Okay, the other one is we're using supramolecular materials to create things that behave like living creatures. Okay, so this is not for medicine. This is actually for energy, for manufacturing, maybe for chemical production, but we want materials that behave like robots, basically. And the Department of Energy is funding our research on this. So this is very new work. You can see this paper was published in 2020 and already into the pandemic. And basically we use chemistry that responds to light, and when it responds to light, it causes materials to shrink or expand reversibly as a result of the chemistry. And so you see here in this next slide a red material that when you shine light from the bottom, this is a red material, and it's in a water tank, because it's a hydrogel, so it looks like an aquatic creature. And when you shine light with an LED from the bottom, it changes shape like that. If you do it from the top, it closes up like a flower, as you see here. And then, very excitingly at the bottom, we see the material in periods of light and dark. We can make a macroscopic material, 1 centimeter in dimension. It can walk. So look at this thing walking. It's walking on a substrate. So if I put it here on this podium, this little 1 centimeter thick, and use light, I can make this material go to Maria. Eventually, very slowly, but it will get to Maria eventually. To deliver something to you. Okay. The science behind it is really fascinating, and the paper, for example, with using simulations and coarse-grained, because you can't do it atomistically, is too much computer time, impossible. We don't have a computer large enough. But basically, the supramolecular structures that we put in are able to be a vasculature. They are very good at conducting water, so they can pull water out of the material very quickly and cause it to shrink or expand more rapidly. And then recently, this is a paper from Science Robotics, also in 2020, where we put now nickel nanowires along with the soft material. So this is now a composite of nickel nanowires and soft material, and so that we could get response to a magnetic field, because the nickel nanowires are ferromagnetic. Okay, so I'm going to show you that material. It's yellow. And this material, as you see here at the bottom, that's a schematic. It's just a cross. We cut it. My postdoc, who is a phenomenally talented postdoc, and he just did something very simple. We took the film and he cut with scissors this cross where all the nickel nanowires had been oriented in the magnetic field during polymerization of the monomer of the soft material. Very simple thing. If you put this cross in a magnetic field, the cross doesn't do anything. It just sits there. I mean, it just doesn't do anything. But then we also put the chemistry that is light responsive. So now you see the yellow material is irradiated with light, and it bends just like the red one, just as a result of the light. Then we turn on the magnetic field, and when it is bent, now under a rotating magnetic field, it walks. And it walks with human speed because it is 1 step per second. We humans walk about 1 step per second. So this guy is moving, okay? And so just for comparison, the red one, it took the red one about 24 hours to go a few centimeters. And this guy, in real time, is going in a few seconds, basically, the same distance. So anyway, so this one doesn't have supramolecular components. This was the early version. So now I want you to see, well, first I show you how we are able, we've done all the mathematics here. If you look at the supplementary material of the paper, there's like 20, 30 pages of mathematics where we actually calculated the magnetic torque, and the model predicts everything. So we can actually program the objects to move. This is experimental, and this is simulation based on the mathematical work that we did. And you see that we can actually predict what the trajectory is going to be. So we can program, in fact, in our lab, what we do is we have a computer, and my students program the little robot to go a certain trajectory, and then it walks, makes a turn, and now it's going to make a turn again, and it's all driven by the magnetic torque up against the soft matter that's around the wires. But now, look what happens when those same materials become partly supramolecular, which is the subject of my lecture, right? So now I'm going to show you the first bend of that yellow thing was accelerated 20 times, so it's kind of slow, okay? Now I'm going to show you real time. And you're going to see on the left, you're going to see the material without supramolecular component, and on the right with supramolecular component. This is real time. Look at the agility. Agilidad se dice en español. Look at the agility of the one with supramolecular structure compared to the other one. This is real time. So the supramolecular nanofibers give agility to this piece of soft matter. We're very excited about that. We're making interesting things. For example, this thing can capture a ball, like a cargo, and then we can put it in the magnetic field and make it rotate. It go to another place, and then when we're at the other location, we expose it to light, and then this thing unbends because we shine the light from the bottom, and then it releases the cargo. So this is a prototype to show, and of course what we're going to do next is we're going to make these things microscopic. So they'll be like the size of cells. So there'll be another, I guess this is really synthetic biology in a real sense, not synthetic biology as the biologists call synthetic biology. But anyway, so we're excited about that. And there's one recently, this is unpublished, but that we figured out how to make them swim instead of just walking, because the other ones don't swim. But look at this one. This one swims and is able to swim because of the supramolecular component. And you see that we put a dye in the water so you can see the convection happening as this thing swims. And then we have other things that change shapes with light. Okay, we have things, very, materials that can walk this way, and they're made up of segments of light responsive things. And this is a very new paper from this year, earlier this year. And so we can, this is like, so the idea is bringing living creature behavior to materials. So they behave like they're alive, but of course they're inert. They're not alive, but you can make them move like living things, and eventually we will put a brain in them by making the molecules and the assemblies more sophisticated. Okay, and the last thing I will mention is that all of this scheme is being used in our lab to develop artificial photosynthesis material. So we have this supramolecular material, this red thing, that makes hydrogen internally, makes hydrogen fuel as a result of light exposure. We very recently showed the critical supramolecular polymer that harvests the light and makes the hydrogen form. And one that I'm very excited about is this one, where we have a new material, which you see it here in bright green, because it's fluorescent, that actually is able to make hydrogen peroxide under light. So this is our effort, basically, to make materials that are like the leaves of trees. The leaves of trees, what do? They use energy from the sun, right? And water, right? And CO2, and they make food, right? So here, the material is like a leaf, literally like a leaf, because it's a hydrogel. It's like a leaf material. And inside is very porous and all of that. And inside, when you expose it to light, it makes something useful. And hydrogen peroxide is very exciting because it's potentially, in contrast to hydrogen as a replacement for fossil fuels, hydrogen peroxide is a liquid. So we could have a liquid fuel, which is less dangerous than dealing with a hydrogen fuel. So I'm sure engineers would be happier with liquid fuels than with hydrogen. Okay, so just, I think I've said enough, and probably this set of conclusions just summarize, you know, the journey that I tried to take you into with this lecture tonight. And I'm sorry that I took more than what Maria was hoping, but I'm very excited to be here tonight, and I just wanted to enjoy interacting with you and giving you the story. So the last thing is a picture of my lab, because without them, it's impossible to do anything. And I want to show you Thaida in the picture. So there she is, and so she is right there. And that's Zaida Alvarez Pinto. And then I have also another Spanish ex-member of the group, Ivan Sacelli, who is now in Biomagune in San Sebastián. And Zaida actually is in the Institute of José Samitier, who's sitting right there in the first row. She is a new staff member in IBEC in Barcelona. Okay, so again, thank you once more for the honor, for the recognition, for the friendship, and my very best wishes to the Royal Academy of Engineering here in Spain. Thank you.
Don Samuel Stupp se acercará a la presidencia para recibir la medalla y el diploma que le acreditan como académico correspondiente de la Real Academia de Ingeniería de España. Creo que está. A ver. Le estoy mirando. Yo también. Esto para ponértelo aquí. Te lo doy. Y este es el diploma. Ya te puedes volver allí. Te puedes sentar ya como académico. Te puedes sentar y ya, sí, esto y eso guardamos. Sí, te lo llevo. Te lo guardamos. Pero ya eres académico, te sientas. Y una vez que ha ingresado ya, quiero decir que hemos celebrado esta reunión. Ha sido interesantísimo. Marita, tenía que haberte dado más tiempo, por todos los que estabas cantando, podríamos haber estado horas. Creo que lo que estás haciendo, o sea, es trascendente, o sea, afecta a todas las tecnologías, afecta a medicina, ¿no? O sea, que creo que son unos temas fundamentales. La ingeniería puede ayudar a la sociedad, no somos mentes cuadradas que nos dedicamos a grandes obras. La sanidad, la medicina, está muy apoyada por lo que hacen los técnicos y los ingenieros. Y este caso, el vuestro, también lo que hace Marita, es la demostración. Hemos celebrado la reunión aquí porque normalmente las ceremonias de ingreso de académicos se celebran aquí, pero debido a todas las condiciones especiales de la pandemia, que no podemos ser muchos, no sé qué, habíamos pensado celebrarlo en los salones de arriba, que luego os enseñaremos, que sería más bonito, más vistoso, pero el sitio donde leen los discursos los académicos es aquí, para que en las condiciones especiales, que incluso hoy a lo mejor no llegabas en el avión, ¿no? Ha habido momentos que no sabíamos si iba a llegar, lo hemos querido celebrar aquí. Pero ahora, como somos pocos, los que tengan tiempo, podemos hacer una visita a la academia, al Palacio de la Academia, que es muy importante, y parte de la historia de España ha sido aquí, en este palacio, por los antiguos propietarios del palacio, ¿no? Entonces, muchísimas gracias a todos por haber acudido, muchísimas gracias a ti, es un honor y un prestigio para nuestra Academia Española tenerte a ti, presumiremos de nuestro amigo Samuel. Yo estudié también en una universidad que se llama UCLA, ¿no? ¿No hay una universidad que se llama UCLA? UCLA. Luego hablamos, yo estudié ahí también. Luego hablamos. Entonces, muchísimas gracias a todos, queda clausurada la sesión y ahora a los que queráis, damos una visita a Palacio. Muchísimas gracias a todos.